Hi, my name is Stephen Leonard. It is December 10th, and this is my presentation on determination of gender, genes versus hormones for the physiology course. So everything starts with genes. Based on gender, the typical individual either has a Y gene or does not. When an embryo reaches 7 weeks of development, the presence of the Y chromosome and the sex-determining region of the Y chromosome, or the SRY gene, initiates a series of developments that results in either the male or female characteristics being developed in an embryo. Before a gender is determined, the embryo contains a bipotential gonad. The structures will become either male or female reproductive tissues depending on a pathway of genes and hormones. This gonad consists of 4 major parts: an outer cortex, which can become ovarian tissue, and an inner medulla, which can become the testes. Wolffian ducts, which stay active in males, and the Mullerian ducts, which remain active in females. As we can see from this image, depending on the presence of the SRY gene, either male or female characteristics will arise from this one gonad. So let's concentrate on the male development. Male development occurs in the presence of the SRY gene and the Y chromosome, which produces a protein called the testes determining factor, or TDF. This protein binds to DNA and initiates the manufacture of SOX9, Williams tumor protein, and steroidogenic factor. These genes and proteins they produce direct the development of the inner medulla of the bipotential gonad into the testes. The SRY gene and these proteins take us through the first 3 steps of the image shown here. The branches of the male development are controlled further downstream by hormones produced by the testes that these proteins help to create. There are 2 types of cells that produce hormones from the testes in this stage of development: Sertoli cells and interstitial cells. Sertoli cells produce anti-Mullerian hormone, or AMH, which does exactly what it says. Remember the Mullerian ducts mentioned that contribute to female development? This hormone initiates the regression of those ducts and inhibits the formation of female characteristics. The second type of cell, interstitial cells, produce 2 types of hormones: testosterone and dihydrotestosterone. Testosterone motivates the Wolffian ducts to become male accessory structures such as the epididymis, vas deferens, and seminal vesicles. And it later initiates the migration of the testes into the scrotal sac. Dihydrotestosterone begins the differentiation of the external genitalia into male sex characteristics. Next, we'll focus on the cause of the female development. I started with the male development for a reason, though, that being that the male development is dictated by the presence of specific genes and hormones created by the Y chromosome and the SRY gene, and that the female development is characterized by the lack of these same genes and hormones. A bipotential gonad will develop the females if there is no SRY gene present. No SRY means no testes determining factor, and no anti-Mullerian hormone. In the absence of these, the bipotential gonad outer cortex develops into ovarian tissues, and the Mullerian ducts develop female accessory structures, like the upper portion of the vagina, the uterus, and the fallopian tubes. Without testosterone, the Wolffian ducts degenerate, and without dihydrotestosterone, the external genitalia take on female characteristics instead of male ones. Together, the presence of Y chromosomes and the SRY gene and the hormones produced by its influence, or the lack thereof in the case of the female gender, determine the gender of an embryo. Thank you.